Bismillahirrahmanirrahim, and welcome back to this next video. And this is the first video in the series of videos on the insulin signaling pathway. Now, in this video and the videos to come, I'll be focusing on the receptor that is involved in the insulin signaling pathway. I'll be focusing on the signal, which of course that would be the insulin, and then we are going to focus in detail on the signaling pathway, how the insulin signaling pathway is responsible for the glucose uptake by the cells. Of course, insulin do a lot of things, but our main focus would be how the insulin is responsible for the glucose uptake by the cells. And when you talk about the insulin signaling pathway, you are focusing about a pancreatic hormone, which in this particular case would be the insulin. Now, why this is important? Why understanding the insulin signaling pathway is so important? Uh, when you understand this complex molecular cascade, you are going to be understanding the glucose homeostasis, because the insulin signaling pathway is very crucial for regulating the glucose homeostasis in the living organisms, for example, the human beings. They are very important for protein synthesis. They are also very important in the metabolism of the lipids. The insulin signaling pathway, if you understand that, that means you want to understand, or you are going to understand the diabetes, the obesity, and some other metabolic disorders. And understanding the insulin signaling pathway is very important for the drug development, because you can identify targets for drugs when you understand the insulin signaling pathway. The insulin signaling pathway is also very important for cell growth and proliferation. And in cancers, this is very important, how the insulin signaling pathway that is not working properly. And in the end, if you are going to understand the insulin signaling pathway, that would mean you are going to have genetic insights. And these particular genetic insights are very important in understanding the diabetes, or for the development of the drugs for the diabetes. So, let us talk about the receptor. Now, in my previous video, I've told you that whenever you talk about a signaling pathway, there would be a receptor and there would be a signaling. So, in this particular video, I want to focus on the receptor, which is known as the insulin receptor. You can call this is the insulin, and then this is going to be the receptor. Now, this is a very general diagram of the insulin receptor. If you look at this receptor, you are having two alpha subunit. This is one alpha subunit, and this one is another alpha subunit. So when you talk about the insulin receptor, you are going to have two alpha subunits, and then you are going to have two beta subunits. So this is one beta subunit, and this is another beta subunit. So that would mean you are talking about a tetrameric molecule, two alpha subunits, two beta subunits. That would mean you are having a tetrameric molecule. Now, let us first talk about the beta subunit. Now, the beta subunit have different specialized domains. Now, the part of the beta subunit that is outside the cell membrane, now these two lines, they are the cell membrane. So, if you talk about this one, that particular part of the beta subunit, which is outside the cell membrane, that is known as the ectodomain. It is also known as the extracellular domain. So, this is going to be the extracellular domain, starting from here, moving up to here. Then you have got the transmembrane domain. So starting from here or up to here, yeah, or you can say that the part of the beta subunit that is in the cell membrane, that is known as the transmembrane domain. And then you have got the tyrosine kinase domain, which is represented over here by the TK. And this is known as the tyrosine kinase domain, which is going to be phosphorylating itself and the other substrate. We will see that in the pathway, how the tyrosine kinase domain it works. And of course, then you have got the carboxylic domain. So this is one beta subunit. This is another beta subunit. So you are actually talking about a tetrameric molecule having two alpha and the two beta subunits. Now, if this receptor is for the insulin, so where the insulin binding site, that is available. So the insulin binding site, also known as the ligand binding region, that is present in the alpha subunit. So these are actually the alpha subunit which are responsible for binding to the insulin and hence triggering the pathway. Now, if you're talking about a tetrameric molecule, what about the structural integrity? How the structural integrity that is achieved? So that is achieved by the disulfide bonds. And if you can see these black lines over here, I'm just using them to give you the concept of the disulfide bonds. So the two alpha subunit, they are connected to each other by the disulfide unit, and at the same time, the alpha and the beta subunit, they are also attached to each other by the disulfide bonds. Excuse me. So these disulfide bonds, they are actually responsible to give structural integrity to this tetrameric insulin receptor. Now, when we talk about the insulin receptor, that is actually an example of the receptor tyrosine kinase. So, what a receptor tyrosine kinase mean? Have already explained that in my previous video, but just to give you a recap, we are talking about a tyrosine kinase. That would mean that this particular in this particular part of the receptor would be responsible for adding a phosphate group to the tyrosine amino acid in its substrate protein. Therefore, this is tyrosine kinase. So, this kinase means it has the ability to add a phosphate group. This tyrosine is actually giving you the name of the amino acid where a phosphate group that would be added. So, that is the tyrosine. So, this kinase is going to add a phosphate group in its substrate protein at its tyrosine amino acid. Now, as this is part of a receptor, therefore we call this is the receptor tyrosine kinase. So this receptor tyrosine kinase, that is, you can say, not a free floating tyrosine kinase, that is part of the beta subunit and hence the insulin receptor. Therefore the insulin receptor, they are also known as the receptor tyrosine kinase. Now let me give you some important information about the insulin receptor over here. Now, the gene that is responsible for the formation of the insulin receptor, that is known as the insulin receptor gene. Now, the insulin receptor gene is located on the chromosome number 19, on the short arm of the chromosome number 19, and a band and a subband position of 13.2. So you can write the locus of the insulin receptor gene as 19p13.2. Now, this particular gene have got 22 exons. From exon number 1 to 11, they code for the alpha subunit, and from exon 12 to 22, they are actually coding for the beta subunit of the receptor. Now, the molecular weight of each of the alpha subunit, that is 135 kilodaltons. The molecular weight of each of the beta subunit, that is 95 kilodaltons. Now, the beta subunit, as I've told you, that have got the tyrosine kinase activity, and the number of the tyrosines that are phosphorylated by this beta subunit, the tyrosine kinase domain of the beta subunit, that are between 6 and 13. And the most frequently phosphorylated positions that have been reported in the scientific literature, so they are at position number 1146, you have got a tyrosine, and that is the most frequently phosphorylated. Similarly, position number 1150, 1151, 1316, 1322. So these are actually the tyrosines, the position of the tyrosines that are most frequently phosphorylated in the beta subunit of the insulin receptor. Now, you might be thinking that the alpha and the beta subunits, they might be different proteins, and they may be coded by a different kind of the genes, but the insulin receptor gene encode a single gene product that is later cleaved and assembled into the heterotetrameric insulin receptor of the two alpha subunits and the two beta subunits. Now, what that means is you are making a very large precursor polypeptide. You are actually going to cleave that to give you the alpha and the beta subunit, and then with the help of the disulfide bonds, you are connecting the alpha and the beta subunit to each other. So the alpha subunit, which of course is the extracellular one that is present, you can say, on the exterior of the cell, and the beta subunit that is spanning the membrane. So they are therefore not encoded by different genes. Instead, they are part of the same protein that are processed into separate subunit after the process of the translation, and you are going to get, you can say, a tetrameric insulin receptor. In my next video, I'll be focusing on the signal, which in this particular case would be the insulin, and we are going to see the structure of the insulin and how the binding of the insulin that is responsible for activating the insulin signaling pathway.